Hello, class. This is the last lecture in the cognitive neuroscience discussion. So we started off with a discussion of the computational theory of mind, and then we discussed connectionism. We discussed whether thinking about the mind as a computer made sense. There's also been quite a bit of advancement in neurobiology and neurology since cognitive science was first formulated in the 50s. And so now we're going to bring that into the discussion. Relevant background is going to be the early neuroanatomy lecture from the beginning of the class, where we discussed the difference between whole brainers and people that thought that parts of the brain were responsible for various facets of your personality and functionality. So both in a sense are true, but it's not the whole brainer portion, which accepted that if you lost 10% of your brain, you'd get 10% dumber, 10% slower, 10% everything. That part is certainly false, but your entire brain is involved in many parts of your life. Also, integration, the integration discussion is going to matter, and mental architectures, this is just a follow-on from that. So this video is probably just going to focus on the first portion here, the cognitive neuroscience. Then we're going to give a particular example with the two visual systems hypothesis, and then raise some problems for cognitive neuroscience. So in the 70s and 80s, we started developing new tools to be able to assess what regions of the brain were affected in particular cases. We'd always been able to see postmortems of brains and see people that had died and that had notorious deficiencies to be able to infer what those deficiencies were and what they were associated with. But surgery got better and better in the 20th century, and so we started being able to make more precise modifications, both of humans suffering from ailments and non-human animals. Actually, to be fair, also humans who weren't suffering from ailments, who were just... the lobotomies were problematic. So I'm going to go through each of these areas in cognitive neuroscience to try to bring the brain back into the picture here, and some of that evidence to see how it fits into what we've been discussing. We're going to start off with discussions of clinical studies, which we're getting better and better and continue to get better and better. Brain imaging methods, again, which the technologies were developed in the past century and have been getting better as we go on. Electrophysiology is a little bit older, at least some portions of it. And computational modeling. Each of these provides different sources of evidence, different kinds of evidence, and how it integrates into the whole field and to our understanding of the mind is going to be an issue. Okay. So we'll start off with clinical studies of neurology patients. Historically, the best evidence we had was indirect. Since the rise of surgeries in the 20th century and our deeper understanding with non-human animals and the surgeries that we do on their brains, we've been able to get more and more precise locations. But the couple of areas we realized were important quite early on were named after these gentlemen, Paul Broca and Karl Wernicke. Broca's area and Wernicke's area, both associated with language. The former seems to be essential in some form to the ability to produce language, such that if you had damage there, you would be unable to produce language, or at least the patients that have had that damage have been unable to produce language. Wernicke's area is the reverse, right? So it's patients who have had damage there have had an inability to understand language. And there is obviously a connection between these in normal controlled patients who can both read and understand, who speak and understand language. There are many famous patients who had particular brain damage. We'll talk about Phineas Gage later on in the semester. We'll talk about a couple of the others. One of the more famous ones is patient HM, whose surgery affecting certain portions of his brain was essential in our understanding of how memory worked. So HM, we now know his name was Henry Molaison, suffered extreme seizures from roughly around the time he was 10 until he was operated on in his, I guess, age 31. The operation stopped his seizures, so he was at least able to keep going with life, but he had an inability to form new memories after this. His life story is fascinating and troubling. The researcher that had access to him also became his legal guardian. There's just a lot of issues here that get into research ethics. Lots of papers were published on him for a long time about how he seemed unable to form new memories and how it seemed like the hippocampus must be essential to memory because he had particular damage there. It turned out that the damage that the surgeon reported was not quite the same damage that we detected in his post-mortem autopsy. So a lot of the papers published between 1957 and 2008 might need to be reassessed. But nevertheless, it showed that memory didn't work the way people would have thought back in modernity. Right? He had damage where he wouldn't remember solving crossword puzzles, but the next day he would be able to do it better than or quicker than before, things like that. So there was something going on, some processing going on. Apparently a genuinely good person. There's a biography of his that I read a couple of years ago, written by the grandson of the surgeon who did the operation. This surgeon also apparently operated on his wife, which the author discovers in the course of writing the book. So it's well worth reading. Anyway, those are the areas that are damaged. Other aspects of cognitive neuroscience that are important are brain imaging, right? So we can actually see what the brain is doing in healthy controls, right? Well, and brain damage patients. We can just see in living patients things that are going on in the brain. So PET scans and functional MRI scans use quite a bit of math and particular techniques using radioactivity and magnetic fields, X-rays to be able to detect certain processes going on in the brain. So PET can figure out which regions of the brain are accepting and requiring more blood flow. fMRIs relatedly can measure this this blood oxygen concentration, both of which seem important, such that if a portion of your brain is being extremely active, it's going to require more blood and oxygen is going to be used much more often. So there's certain assumptions here about cognitive activity and cellular activity and blood flow, or blood oxygen supply. And again, we know that there's lots of other stuff that's happening in the brain, right? We know that there's going to be some interesting chemistry going on. We know there's other stuff going on, but these are the tools that we have that allow us to non-invasively measure what's going on. And non-invasive is good because of ethics is one portion, one reason it's important. But also, you want to non-invasively figure out what's going on, because causing damage to the brain might change the underlying effect. And so that's what we're going to get to with some forms of electrical recording, which is another approach that is common. So humans are biological creatures whose activity produces electrical activity that is noticeable, that is detectable. So Angela Musso did certain studies on this famous patient Bertino to try to see whether he certain activities caused more brain activation. So the forearm is, you know, we would call it a control now. And the idea was to see whether more electrical activity was going on in the brain if Bertino was resting versus hearing church bells versus thinking about saying prayers after the church bells ring versus doing certain mathematical functions. And so people realized that there was electrical consequences of brain activity. So like the blood consequences and the oxygenation consequences, we figured we could start trying to measure this. And so now we have quite a few ways of measuring the electrical output of the brain. So there's a couple of really cool videos online where you can see these TMS, these magnetic stimulation, where a person will put this device on their head as they're speaking and zap themselves, and then they'll stutter in the middle of a sentence or whatnot, as they're disrupting certain parts of their brain by running electrical currents through it. So if we can disrupt parts of your brain and get different results, it seems like those parts of the brain, by assumption, are required for those activities. There's EEGs and ERPs, which you may have heard of. It basically involves attaching electrodes or measuring how particular events affect electrical conductivity. And then a little bit more invasively is the single unit recordings, where we can actually go into the brain and into the body and measure single neurons and their activity. We tend to do this mostly on non-human animals. So the final thing that I'll discuss here in this video before we move on is this idea of computational modeling. So there's two approaches. Well, there's many approaches, but two ways of dividing them you might think of in terms of functional fidelity or cognitive fidelity, and biological fidelity or anatomical accuracy. You can model, so Arguably, that's what a connectionist system is, right? It is a brain model of what's going on, but you can get more or less accurate, more or less involved, and the models could require more plausible or less plausible assumptions, all to try to figure out what's going on in the mind. I'm not going to get into the details of these particular models, but at some point in the class, we'll talk about the Blue Brain Project. It's fascinating. Okay, so we talked about varieties of cognitive neuroscience: clinical studies, brain imaging methods, electrophysiology, and modeling. And you'll notice that I underline and bolded the different questions that are going on here. So what is being affected, where in the brain something occurs, where activity is occurring, where, how something could happen. These are all different questions. And we'll get to how to think about them all together in the next video, in the third section of this lecture. Okay, I'm going to stop here, and then the next video we're just going to talk about the two visual system hypothesis and some problems for cognitive neuroscience.